Hello, we are the Dundee team from Scotland for the 2015 International Genetically Engineered Machine Competition. The iGEM competition draws teams from all over the world to compete in the design and creation of hundreds of different biological devices to carry out a plethora of real-world applications using what is known as synthetic biology. But what is synthetic biology? Synthetic biology, put quite simply, applies the principles of engineering to the field of biology. However, it's not just biology that's involved. Many different disciplines such as mathematics, physics and chemistry are combined in the creation of these biological devices. Examples of devices designed by past teams include glowing bacteria and flavoured yogurts, which are flavoured by the bacterium. But the competition has also seen the production of diagnostic tools for diseases such as cystic fibrosis, achieved by last year's Dundee team. This year, our project has involved applying the concepts of synthetic biology to the world of forensic science. But what is forensic science? It's a study of evidence using scientific methods to determine what happened at the scene of the crime. The evidence obtained is then presented in a court of law to persuade the jury if the accused is guilty or not guilty. However, there's one big problem currently facing forensic science, and that is that it's not particularly, let's just say sciencey. While DNA analysis is a useful tool in investigations, other components of forensic science such as blood detection and fingerprint lifting is not very accurate. For example, fingerprints are often lifted using superglue, a method found purely by chance. And luminol, the substance used to detect blood, frequently yields false positives for fluids such as bleach. Using synthetic biology, we aim to create a set of devices known as the Forensic Toolkit that will be used to improve the efficiency by which crime scene investigators detect and analyse evidence. These include a body fluid detector, a method for fingerprint aging, and a stainless steel detector. The first of these is the body fluid detector. The body fluid detector will glow a different colour according to which body fluid it has detected: blood, semen, nasal mucus, or saliva. But how is this synthetic biology? Well, we are using the bacteria E. coli to act as a factory to produce our detector molecules that will be used to target specific components which are either unique or highly abundant in the four body fluids. However, some of the genes for these molecules are not normally found in E. coli, so we have taken genes from other species of bacteria, mice, and even humans and put them into E. coli. For the fingerprint aging, we are also using E. coli to produce our detector protein, which will target a component of fingerprints whose presence we can measure against time. Our device aims to detect fingerprints that are less than 1 week old, distinguishing them from fingerprints that are months to years old. Our final device, the stainless steel detector, is a bit different and will be applied to bone samples. Some special bacteria can detect a component of stainless steel called chromium, which is found on many metal weapons, such as knives. We have exploited this to make the bacteria glow bright green when they come into contact with it. However, since we need the cells to do this task themselves, we would need to take a sample of bone for analysis back in the lab, as the cells could leave traces of DNA. The stainless steel detector is different from the first two devices, since in their case the detector molecules would be harvested from the bacteria and used in a cell-free manner to avoid contamination of crime scenes with foreign DNA. So what do we hope to achieve? We want our body fluid detector, fingerprint aging device, and stainless steel detector to act as proof of concepts for new applications of synthetic biology. We hope to inspire forensic science to base itself on solid, more science-based foundations and allow for accurate representation of evidence obtained at a crime scene. Ultimately, this will hopefully ensure that justice can be served confidently and categorically.